Okay, so now we're going to start photosynthesis. Photosynthesis is essential to all life on Earth. It's the only biological process that can harness the sun's energy and convert it into a usable source. It's also a source of oxygen for all living things, so without photosynthesis we wouldn't be able to breathe. A lot of the energy extracted today from the fossil fuels that we use represents captured light energy, and it's about 200 to 350 million years old. So there are a couple of different types of converters of energy we want to talk about. So photoautotrophs are going to use light to manufacture food. Examples of this are plants, algae, and cyanobacteria. These are the only organisms that are capable of photosynthesis, or this transfer of light energy into chemical energy. Heterotrophs rely on sugars from photosynthetic organisms for energy needs. This is like us, and animals, and fungi, and most other bacteria. There's also a really specific and strange type of bacteria called chemoautotrophs that synthesize sugar from inorganic compounds. Photosynthesis is extremely important. It's going to store energy from solar radiation in carbon-carbon bonds of our carbohydrates. And carbohydrates are the energy source of ATP production, so this powers about 99% of Earth's ecosystems. Photosynthesis requires some specific wavelengths, so it will require a talk on light energy, which we will talk about, carbon dioxide and water. So it's a symbiotic relationship with us and other animals that are going to release carbon dioxide as a byproduct of our metabolism. And it will produce oxygen, glyceraldehyde 3-phosphate, and simple carbohydrates to convert into sugars for energy. So photosynthesis, these structures that create or undergo photosynthesis, it takes place in plant leaves, specifically in the mesophyll, which is the middle layer. Gas exchange happens in stomata, which are these little holes that lie on the underside of the leaf. These help to minimize water loss due to high temperatures. They can open or close due to osmotic changes. So within the mesophyll, photosynthesis will take place within the chloroplast, which is an organelle. This chloroplast is derived from ancient cyanobacteria, so it's an organelle that's derived from an organism. Pretty crazy. Within the chloroplast are these stacked discs called thylakoids, and within them is chlorophyll, which will be our pigment that causes the green appearance of plants. So photosynthesis takes place in two stages. We have light-dependent reactions, which, as you would expect, require light. And stage two is light-independent reactions, so it doesn't require light. So in stage one, the energy absorbed by the chlorophyll will be converted into chemical energy, and then energy carriers will store that energy. In stage two, that chemical energy will be used to make sugar. So here's that light energy talk we were talking about. Humans can only see a small fraction of light that exists. We can only see what's called visible light. Wavelengths are used to determine the amount of energy from a wave of light. The longer the wavelength, the less energy it carries. So shorter wavelengths tend to be more powerful. Wavelengths can be measured by crest to crest of each wave. The electromagnetic spectrum is a range of all possible frequencies of radiation, so all of the light that is possible. Each type will emit a different wavelength. So pigments, when we start thinking about colors, are going to absorb wavelengths of visible light. However, they reflect the wavelengths that they cannot absorb. So the wavelengths that they cannot absorb, that are reflected, are the wavelengths that we see in visible light. Our retinal pigments range from about 700 nanometers to 400 nanometers on this wavelength spectrum, and that's about where the photosynthetic pigments lie as well. We can also see sometimes with white light, a prism can disperse it into different colors, which is what happens with our vision. So in plants, we have some different types of pigments. We already talked about chlorophyll, but there are multiple types of chlorophyll, A through D. Chlorophylls A and B are found within our plant chloroplasts. There's also carotenoids, which are found in fruits and vegetables, and they're really good at disposing excess energy as heat. Pigments can be identified as by wavelengths of visible light. So like we said, pigments are going to absorb certain wavelengths of light, but reflect wavelengths that they cannot absorb. So chlorophyll A absorbs blue and red, but reflects green because it cannot absorb it. Spectrophotometers, these really cool machines that we weren't able to use in lab this semester, they differentiate wavelengths of light a substance can absorb. So they measure the transmitted light that is reflected from the pigment. Okay, so how does this pertain to photosynthesis? We're going to use this solar energy to create ATP and NADPH. That's the short of it. We're going to do so by photosystems. So we have two photosystems, Photosystem 2 and Photosystem 1. I know that that's weird that they're named backwards, but they were named based upon when they were discovered, not on what order they go in. So photosynthesis occurs here. Both photosystems are going to contain these antenna proteins where the chlorophyll will be bound, and they surround a reaction center. Both are serviced by something called a light harvesting complex, which is going to pass energy from sunlight into the reaction center. An absorption of a photon of light energy will send this whole complex into an excited state. That light energy will be transferred from chlorophyll to other chlorophylls, but it's not usable just yet. When chlorophyll A gives up an electron, that's called a photoact. That electron is then carried by NADPH to the Calvin cycle, which will be our light-independent reaction. So we'll use those electrons later. Photosystems 2 and Photosystem 1. Both Photosystem 1 and 2 are major carriers in the electron transport chain in plants. So each reaction center of the photosystems will have a different name. So the reaction center of Photosystem 2 is called P680. It's going to deliver electrons to the primary electron acceptor in Photosystem 1. This process will split water to release oxygen as a byproduct, which is either used in the electron transport chain or by aerobic organisms like us to breathe. Hydrogen atoms are going to be used to synthesize ATP, just like they are in our electron transport chains. Energy from the electrons are then sent to the Photosystem 1 reaction center, which is called P700. This will send electrons to NADP+ to reduce it into NADPH. Photosystem 2 will capture the energy to make ATP, and Photosystem 1 will capture the energy to convert NADP+ to NADPH. So just like in our electron transport chain, the buildup of hydrogen ions will create this concentration gradient, and the passage of ions from this high to low concentration is what's going to form our ATP. And it's the same process of chemiosmosis via ATP synthase. So the Calvin cycle, like we said, is our light independent reaction. That carbon dioxide will enter the leaves through the stomata on the undersides of the plants and enter the stroma of the chloroplast. There's 3 stages: fixation, reduction, and regeneration. So in this cycle, we're taking carbons off of these carbon dioxide molecules to make sugars, and the O2 that is left over is released as a byproduct. So in stage 1, we have fixation. We have an enzyme that has a really long name, but we abbreviate it to Rubisco. We have 3 molecules of RuBP and carbon dioxide. Those 3 things are going to initiate the reaction. This catalyzes the reaction to form 3-PGA. And in this step, since it's called fixation, we're taking those carbons from carbon dioxide and we're fixing them, aka changing them from the inorganic form of carbon dioxide into the organic form of a carbohydrate, sugar. In stage 2 is reduction. ATP and NADPH are used to convert 3-PGA into G3P. Electrons are gained via the reduction reaction. 6 molecules of ATP and NADPH are used in this process. So remember, in the light-dependent reactions, we're making ATP. In the light-independent reactions, we're using this ATP to make sugars. ADP and NADP+ return to the light-dependent reactions to be regenerated as ATP and NADPH as usable energy, so that it can come back into the Calvin cycle and this cycle can continue. In stage 3, we have regeneration. 1 G3P molecule will leave the Calvin cycle and enter the cytoplasm, and this will help in the formation of other needed plant compounds other than just these sugars. The rest of the G3P molecules will remain to regenerate RuBP to fix more carbon dioxide into these long carbon chains of carbohydrates in simple sugar form. So, a recap of photosynthesis in electron transport chain, but in a plant. So, instead of all of our complexes of complex 1 through 4 and all of our protein carriers, we just have photosystem 2 and photosystem 1, and they're still just passing electrons through and shuttling hydrogen ions so that they can be used by ATP synthase to make ATP, which is then used in the Calvin cycle to make simple sugars. So all living things access energy by breaking down carbs. We do, plants do, bacteria do, everything. Every atom of matter and energy is recycled. So nothing that we are eating today hasn't already existed before. Those atoms were just part of something else, and they're constantly being recycled as new matter. So photosynthesis and cellular respiration cycle in harmony. We release carbon dioxide as a byproduct, which is used in photosynthesis to use those carbon dioxide molecules in the Calvin cycle to make simple sugars. And then oxygen is released as a byproduct that we breathe in to make more carbon dioxide. So we cycle in continuous harmony with plants.